So people, are you curious to know the top priority questions that come from embryology in the final anatomy theory exam? So to know that, you should look at the last 5 years previous year questions. That's what we're looking at, previous year questions. Let's get started. So without wasting time, the first question here, we're going in order of all this oogenesis, spermatogenesis, etc. So they will ask you spermatogenesis. So you should know how the spermatozoa are formed. So here you look at this, you have the primordial germ cell, then you have the spermatogonium, then you have this primary spermatocyte, which is the largest germ cell. It will undergo the first meiotic division to give you the secondary spermatocytes, which will then undergo the second meiotic division. So you will have the spermatids, and from the spermatids, you will get what, people? Spermatozoa, 22x, 22x, 22y and 22y. So basically you started off with what? 44xy, and you have landed up with 22x, 22x, 22y, 22y. Primordial germ cell, spermatogonium, primary spermatocyte, that is 44xy. Then after the first meiotic division, you will get the secondary spermatocyte. Then you have the second meiotic division, which will give you the spermatid. Okay, spermatid will come after the second meiotic division. And the spermatid will become spermatozoa. Okay, so they are calling this as spermiogenesis. The entire thing is spermatogenesis. Only this maturation kind of thing from spermatid to spermatozoan, they are calling it as spermiogenesis. One question in embryology done. Let's move on to question number. Then coming to diagram of Graffian follicle, you should know, people. Graffian follicle is the mature antral follicle in the ovary, which will release the oocyte, right, during ovulation. So you should know how to draw the diagram. They've asked you to draw the neat label diagram. So this is within the ovary. Here you have the tertiary or the Graffian follicle. So that, when we have shown here, it is looking like this. This you'll have to draw. Now let me zoom here and explain the layers that you should draw in the exam. I'm just giving you a very high level overview here. So here you have the oocyte, around which you have the vitelline membrane, zona pellucida, and the corona radiata. Here you have the antrum, which has the fluid, follicular fluid. Then here you have the granulosa cells. Then you have out of that, what you have? The theca interna. Then you have the theca externa. Okay, so these are the layers that you should know. How's it going, people? We are looking at the PYQ for embryology, right? Focus here. The next question that you should know is fertilization. Basically, fertilization is the fusion of the male and female gametes to produce zygote. So it will decide the sex of the zygote. It will also make the haploid into diploid bag, right? Then what else? Cleavage, initiation of cleavage, you can say. Okay, for this fertilization, you basically will have to know about the capacitation of the sperm, where it will lose the acrosomal cap and all the enzymes, hyaluronidase, acrosin, etc. are released. It is penetrating the corona radiata, the zona pellucida, and then the vitelline membrane, right? So you should know this. Then the how the oocyte is undergoing the second meiotic division here, right? So here you are, they are showing you the formation of zygote. So you can see here they have asked this for 5 marks. So you should know everything about fertilization. Complete chapter you have to read. There's also, they are saying the result of fertilization is the cleavage formation. Cleavage consists of repeated mitotic divisions of the zygote into smaller units. So here they are showing you the cleavage of the zygote. So you are able to see the names of these cells. See, single cell stage, 2 cell stage, 3 cell stage, 4th cell stage, 16 cell stages, morula. Then it comes blastocyst, okay, after that. So at the end of cleavage, what they have shown you is the morula, 16. In some exams, they will ask MCQ also, correct? So look at one of the MCQs. They've asked you the 3 barriers that the sperm has to penetrate before fertilization includes all except. So it will have to break the vitelline membrane, zona pellucida, and corona radiata. So the first thing it has to break is the corona radiata, then the zona pellucida, and then the vitelline membrane, right? So acrosomal cap, it doesn't have to break. That is a part of the sperm itself, the sperm cap. So that is called as capacitation, right? When it is releasing those enzymes, losing that acrosomal cap. Here you have the acrosomal cap. How's it going, people? Shall we go to the next question? You have learnt a lot already, isn't it? Good. Results of fertilization, they have asked for 3 marks, 3 things they want you to write. You will have a zygote, then it will be diploid, then there is initiation of cleavage, then what else? A lot of other things you can write. There is going to be the second meiotic division of the oocyte, right? Then there's going to be variation in the species. Okay, some philosophy you can write. It's not philosophy, but yes, they have written it in the textbook, so you write that. All are the end results of fertilization except. It is an except question, except. So I'll use a red here. So end result of fertilization, you tell me. Restoration of diploid number of chromosomes, yes. Determination of sex of embryo, correct. Completion of the second meiotic division of the female gamete, yes. Correct. So I know which the except answer is, this one. Okay. So the answer here is this one. Okay. So other 3 are the results of fertilization. You should know them. Now moving on to the previous year questions. Describe the development of umbilical cord. Explain why umbilical arteries carry deoxygenated blood and umbilical veins carry oxygenated blood. Okay. So first of all, the development of umbilical cord. You have to explain. I think this is just a little too complicated. Is it? Let's look at it. Umbilical cord is a long twisted beaded like structure by which fetus is attached to the uterine wall via placenta. Okay, so I don't want to spend too much time in this video about all this. So here you have the placenta and here you have the umbilical cord and you have the fetus here, right? This is a fetus, and then here you have the uterine wall, let us say here, uterine wall. Okay. So what is umbilical cord? It is a long, twisted, beaded-like structure by which the fetus is attached to the uterine wall via the placenta. What does it have? It is having an umbilical vein and 2 umbilical arteries, correct? So it has 1 umbilical vein and 2 umbilical arteries in the cross section if you see. So what does this umbilical vein do? Umbilical vein will carry the oxygenated blood from the mother, not the blood, the oxygen. Oxygen it will carry from the mother to the fetus. So this is actually the fetal blood only. So it is carrying this blood, oxygenated blood, but the oxygen is coming from the mother to the fetus. Then umbilical artery is doing what? It is going to take the deoxygenated blood and give it to the placenta. But what have they asked here? The development of the umbilical cord. So basically it develops from the connecting stalk part of the extraembryonic mesoderm. Mesoderm, this is the word that you have to focus on. What is it? Mesoderm. I don't want to highlight the entire thing. Only this mesoderm I wanted to highlight, but I'm not able to highlight because I'm clicking on some wrong button, isn't it, people? I really don't know how to highlight only this word. Okay. So mesoderm, umbilical cord. Then you'll have to draw these diagrams also. We are not going into the details. Connecting stalk part of the extraembryonic mesoderm. It is extraembryonic. It's outside the embryo, looks like. Okay. One more term important here could be this Wharton jelly. The umbilical vessels develop in the connecting stalk. Obviously, inside this connecting stalk only these arteries and veins will develop. And the primary mesoderm of connecting stalk undergoes mucoid degeneration to form a gelatinous substance called Wharton jelly. Okay. So this Wharton jelly is around these vessels and it protects these vessels, looks like. Contents of umbilical cord, you have the umbilical vein and then the 2 umbilical arteries and a lot of jelly. Okay. What jelly? Wharton jelly. So basically, let me explain again. Why is there, what did they ask? Something they have asked about umbilical vein, right? So umbilical vein is going to take the oxygen from the placenta to the fetus, and then once the circulation is done, the arteries, the 2 umbilical arteries are carrying the deoxygenated blood to the placenta. So why is the question if they have asked? You have to say that the fetal lungs are not, fetal lungs are not functional yet. So the oxygen is supplied by the mother. Remember, the blood is not exchanged between the mother and the fetus, just the oxygen. Here you'll have to explain fetal circulation. So if you remember this diagram from the fetal circulation, we've already covered this in a different video. Look at this: placenta, umbilical vein joining the portal vein, inferior vena cava. The blood then reaches the right atrium, directly goes to the left atrium, gets to the left ventricle, gets pumped to the entire system of the fetus. Then it becomes deoxygenated, and then the umbilical arteries carry this deoxygenated blood back to the placenta. This is the fetal circulation. Here, if you want to explain even more, you can say the umbilical vein and the portal vein, right? Umbilical vein and portal vein are joining. Here you have the ductus venosus, right? Then inferior vena cava. From here, right atrium to left atrium, that is via the foramen ovale. Then from the left ventricle to the entire body. Here there is one more shunt between the pulmonary trunk and the aorta, that is ductus arteriosus. Okay. So umbilical cord development they have asked. Remember to explain some clinical anatomy. So when the newborn is delivered, the pediatrician, when they cut the umbilical cord, they will check the veins, right? And the artery. No, we should say they count the single vein, it should be there, and the two arteries, okay? Okay, the next important question for you here in embryology is describe the chorionic villi. Okay, classify. You have to classify also. What types of villi are there to classify? Okay, so first of all, it is embryology, so the development of the villi. So you should know, you have to draw this diagram, which is the maternal part, which is the fetal part, how are they joined, okay, all that you have to explain. Even for umbilical cord, if they ask you, you can draw this diagram. So here you have the maternal side, right, and here you have the fetal side via the umbilical cord. So here you have the layers that you should know. These are the villi, chorionic villi, right? What else have they asked you actually? They've asked you classification. So here you will have to explain that there is something called as primary villi, secondary villi, and tertiary villi. Tertiary villi is this one, which has a lot of blood supply, etc., etc. So this is what you should know. So let's look at the diagram and understand that's better. So the primary villi has what? Cytotrophoblast, syncytotrophoblast that everybody will have. Then coming to the secondary one, it has some extra embryonic mesoderm inside there. And when it comes to the tertiary villus, it has all those three with the fetal capillaries. So the tertiary villi, so the blood vessels will develop in the mesoderm of the secondary villi, right? So once the blood vessel develops, you are calling it as a tertiary villi. The secondary villus with blood vessels in its mesoderm is called as the tertiary villus. So easy. So primary villus has the core of the cytotrophoblast cells covered by a layer of syncytotrophoblast. Secondary has these two with some mesoderm, and then the tertiary or the definitive villus will have the same, all these three things plus the fetal capillaries which are forming in the mesodermal core. Very important to know is the development of the embryo, when what is happening, right? The formation of the primary villi they're saying is day 12. Okay, primary one, 12. So did you classify and describe the chorionic villi for 5 marks? You will have to draw a diagram. Excellent, you have learnt a lot. Continue to learn the embryology. So you always have to give clinical anatomy, clinical embryology, right? What happens if this chorionic villi has some abnormal adherence to the uterus? Okay, so you will have these conditions called as the placenta accreta, placenta increta, and placenta percreta. So this is like percreta is like it is penetrating the full thickness of the myometrium and the perimetrium. So that's really bad, right? It is so adherent. So this also you should know. So this is a question to check if you are awake or not. So please tell us, what are we looking at? Previous year questions under embryology, which have come in the final year anatomy paper. Okay, last 5 years. Okay, embryology. So now let's move on. Define implantation. Mention any 2 abnormal sites of implantation. Ask for 3 marks only. So let's quickly do this. What implantation is? Let's learn that. And then the 2 abnormal sites of implantation. Okay, what do you mean by implantation? So implantation is what happens. The blastocyst, right, will implant. Don't you think that's what implants? So somewhere here it should implant. Or wait, let me use a green. So somewhere here or somewhere here it implants, right? That's what we have learned. So basically implantation is the process by which the blastocyst is embedded into the endometrium of the uterus. It occurs from 6 to 10 days from the fertilization. So where will it implant? So ours is something called as interstitial implantation in all humans. Okay, so humans, we have interstitial implantation. That is the blastocyst. Oops, again the same problem. The blastocyst will embed in the endometrium of the uterine wall. Okay, so I will give some color for this. That's only better because I'm not able to highlight. Endometrium of the uterine wall. Okay, so this is what happens in humans. In others, something else happens. We're not going into that. But that's not what I'm looking for. I'm looking for where exactly in the uterus it should implant. Okay, so I will draw a uterus here. You should draw in the exam, people, and explain what is normal and what is abnormal if you want marks. So very simple, you just draw like this and tell them that the normal sites of implantations is somewhere here, right? So somewhere here. So these are the normal sites of implantation. So how do you explain that? So the blastocyst is implanted in the upper part of the posterior wall of the uterine cavity. So you have to say what? Upper part. Upper only they are focusing on. That the other two things that I told, not that. What they are saying is the upper only. So if this is the uterine cavity, so upper only they are saying here. Okay. So when it goes inside the endometrium, on top you will actually just see a small swelling. Okay. It produces a small swelling on the endometrium. It will go inside and it will implant. Okay. So normal, that is. So now what is abnormal? Abnormal sites of implantation within the uterus are the lower uterine segments near the internal os. That means to say it is like very near to the internal os. So they have shown here. So that is what? Placenta previa it will become. So somewhere here, right? So they are the internal os. So that is abnormal. That is number one they have put. Then you have other things which is outside of the uterus itself. Then it will become more like an ectopic pregnancy, correct? So what are they telling here? Ampulla infundibulum interstitial part of what? Of the uterine tube. That is in the fallopian tube it can implant. So that will become, isn't that same as ectopic? So they are saying in the ampulla, that is here, right? Ampulla is the turning. So ampulla infundibulum interstitial part of the fallopian tube. Are you understanding where I'm marking, people? So let me mark here. So which color shall I use? This one. So infundibulum, ampulla, which is that the bend, and the interstitial part of the fallopian tube. On the other side also same thing. Okay, remember. So these are the abnormal implantation sites of the blastocyst. Very good. The abdominal cavity, forget all this. It can go into the abdominal cavity. They have shown here one of it. Here they have shown, right? Are you able to see? Then it can implant in the ovary also, it seems. What is this? It can implant in the ovary. Fertilization and implantation may occur while ovum is still within the ovary. Oh, interesting. So very crazy things. Okay, all crazy things are possible. Okay, so you learned the abnormal sites of implantation. Good. So what is it if it is near the internal os? Okay, that is the placenta previa. So in that you have the first degree, second degree, third degree. So basically the outlet of this fetus itself is blocked. So you should understand it will have to break through that and bleed because it's the placenta full of blood vessels, right? So it will bleed out. So you'll have to do a cesarean if it is a placenta previa. Okay, then you have ectopic pregnancy. We already told you it is nothing but ectopic because it is outside of the uterus, the normal uterus. So you have the tubal pregnancies, that is the fallopian tube. Then you have the abdominal, we told you, and then the ovarian. Okay, so we have already told you this. So what happens in ectopic? They are not in a favorable location. So the embryo will die, and it can also cause a lot of problem to the mother. It can rupture. It can also lead to the death of the mother. How is it going, people? Now let us go to the next question. Okay. So what implanted? Blastocyst. They want to know about the blastocyst. So they want a neat labeled diagram of blastocyst. So blastocyst is at what day stage, people? What? They're asking diagram only, okay, for 3 marks. But I'm asking you at what day is it? Day 6, day 5, day 6 around, right? So the implantation of the blastocyst happens. So they want you to draw a diagram of this blastocyst. Okay. So blastocyst is the one that implants. It is beyond which stage? It is beyond 32 cell stage. Okay, so focus here. 16 was called as what? Morula. 16 cell stage was called morula. If it is beyond 32, then it is called as blastocyst. Okay, so this is the one that implants, okay, into the uterus. Now let us go back here. We have to draw a diagram. So the diagram you have to draw is so simple. Just draw one circle and put some cells here. This is the inner cell mass. Inner cell mass. This is the inner cell mass. Here you have some cavity of the blastocyst, and here also you draw one line of cells like this. Okay, I'm just trying to roughly draw. So this is your blastocyst. The outer one, whatever you're drawing here, is the trophoblast. This one you're calling as inner cell mass, and in the middle some cavity. Okay. Further, what is happening? You are having endoderm, then you have ectoderm, endoderm. In the middle, you should have some cavity, amniotic cavity. Then I'm expecting the mesoderm to form somewhere, right? So then here you have the NotoCord, etc. will come next. Okay. So if you want to know this, the most important question of embryology, that is this one. That is NotoCord. Yes. So NotoCord is the most important question. It's been asked like multiple times. So let us look at this. What is this NotoCord? First of all, you should know what is NotoCord. You should know it forms. In us actually what it finally becomes is just the intervertebral discs, okay, and the apical ligament of dens. This is what finally it becomes. But why is it so important? Because all animals of phylum Chordata will have this NotoCord. Okay, so here they've shown the NotoCord, right? So basically all animals belonging to phylum Chordata has this NotoCord. Okay, so what does this finally become in us? In us, it will just become intervertebral discs, that to the central part, that is the nucleus pulposus of the intervertebral disc and the apical ligament of dens. That's what it will finally become. This NotoCord for us in humans, it appears only in embryo. Okay. Other thing you should know, people focus, why is this NotoCord so important, right? Because the neural tube is formed from the ectoderm, which is overlying the NotoCord. Neural tube is formed. Okay, nerve neural tube is formed by what? It is formed by the ectoderm. Ectoderm which is overlying the NotoCord. That is why all this so much NotoCord, very important. So which of the following is a derivative of NotoCord? People, you know the answer, you say it. Is it an artery? Is it some venous plexus? No, it is intervertebral disc. That is the central part. That is the nucleus pulposus. So if this is some intervertebral disc, the central part of it, that is the nucleus pulposus, is actually formed, is the derivative of NotoCord in humans. Next question. Mention the formation and fate of NotoCord. From where is this NotoCord formed? So it is formed by from that Hensen node. Okay. These are some terminologies which may come as a question in the exam. See, Hensen's node they have asked. Means what? NotoCord only they want you to explain. See, NotoCord has been asked so many times. Even in this paper they have asked the formation and fate of NotoCord. So around day 16, so the NotoCord forms during gastrulation, which is critical stage in embryogenesis. Basically, from where is it forming? From that Hensen's node. I told you what is this Hensen's node. It is a primitive knot, KNOT knot of the primitive streak. That is all you can say. People, it is so complicated to understand. You just go through some very good YouTube videos. They'll explain. So here you have the primitive streak. So from the primitive streak, right, you have something like the NotoCord. This primitive knot is nothing but the Hensen's node. So you have this NotoCord that is going like this. So the NotoCord develops from the primitive knot, that is the Hensen's node of the primitive streak. So how does it form? A depression appears in the center of the Hensen's node called blastopore. Then what will happen? These cells will grow cranially from the bottom of the blastopore between the ectoderm and the mesoderm up to the prochordal plate to form the NotoCord. So all these terminologies you should say, it is from this blastopore it will grow cranially, okay, between the ectoderm and the mesoderm. So are you able to see all that? So it is growing cranially towards the head, right? from this blastopore. If you can draw diagram good, see from blastopore it is coming cranially, it is growing here between what, that ectoderm and mesoderm, something they said, right? So here you have some canal, it has formed now. Now it is, this bottom part is fusing with the endoderm. Fusion of floor of notochordal canal with endoderm, something inside, it is fusing with that endo. Okay. And then it is communicating with yolk sac. And finally, you have something called as the definitive notochord. So finally, what formed? The notochord formed. From that notochordal, chordal, it became finally definitive notochord. Okay. So why is this so important? Because it forms the central axis of the developing embryo. So here, if you have this embryo, it will form the central axis of this developing embryo, and above this, what will happen? There is a ectoderm, overlying ectoderm is there. From that ectoderm only, that neural tube will form. Okay, so that is why that is very important to know. Okay, so neural tube will form from this overlying ectoderm, which is overlying what? Overlying this notochord. Okay, and basically this will provide a central column, okay, around the vertebral bodies. Okay, so I'm thinking something like spinal column. What do they mean by central column? So basically, clinical anatomy you should know, people. That's why they do in pregnant women NT scan to find out neural tube defects, etc. And then when do you do it? Around 12 weeks, 11 weeks, 12 weeks of pregnancy. You should not miss the date, right? And then chordoma can happen. There's a tumor that can form chordoma from the remnants of notochord. So try to draw this diagram, at least some diagram, like you draw like this and you show some primitive streak, primitive knot, Hensen's knot. From that you show notochord towards this prochordal plate. Okay. Look at the next question here. Classify intraembryonic mesoderm, enumerate its derivatives. So here they are asking you about mesoderm, which mesoderm, intraembryonic mesoderms, and they want to know its derivatives. So this is for 5 marks. They have asked you in final final anatomy theory paper. Okay. So when you say intraembryonic mesoderm, is there something called as extraembryonic mesoderm? So basically this is something like a very different concept. Okay, so this is some kind of special mesoderm they are talking about. So it is the middle layer of the trilaminar embryonic disc. It is the middle layer. I am not going to have any doubt on that. It is the middle layer, right? It lies between ectoderm and endoderm. That also I understand. Except in the following 3 sites. Then where does it lie? If it is not in the middle of these two, then where does it lie? That is in these 3 sites. That is the prochordal plate, notochord, and cloacal membrane. At these places, it lies on either side of the notochord. So this is what is very special here. It lies on either side of the notochord. So let us look at the diagram and try to understand. So here they are telling you that here where are the 3 areas? One is this cloacal membrane, the other one is notochord, the other one is prochordal plate. Here it is not between those 2 things, that is the ectoderm and endoderm. Okay. Here in this diagram they are trying to show that. So here they are showing you neural tube which is overlying this notochord. Okay. And on either side you have this mesoderm, that is the intraembryonic mesoderm. Again in that you have the 3 parts. That's what they are saying. What are the 3 parts? You have the paraxial, then you have the intermediate, and then you have the lateral. So easy to understand, right? Lateral means outside, paraxial means around this axis, that is around this neural tube, and then intermediate which lies between this lateral and paraxial, correct? In between this intermediate. So these are the 3 subdivisions of this intraembryonic mesoderm. So this 2 sides, what will happen? The 2 sides you have that mesoderm, right? Those 2 will join will meet in front of the prochordal plate to form cardiogenic plate. Okay, so these 2 which were here will join in front of the prochordal plate to form something called as the cardiogenic plate. Okay, if you want to remember that you can. Anyways, now let's read this. So what are the subdivisions of the intraembryonic mesoderm? Intraembryonic mesoderm on either side of the neural tube divides into 3 longitudinal columns. We already told you. That is the paraxial mesoderm, that is medial, that is paraaxial, that is on the other side, either side of the axis, that is the neural tube. Then here you have the intermediate mesoderm in between the paraxial and the lateral, and then laterally you have the lateral plate mesoderm. What these actually become, no, that they're explaining here. Okay, the paraxial mesoderm. So where is it? It's beside this neural tube. So neural tube is here, means beside it there is something, right? So what will it form? This paraxial will form the axial skeleton. So vertebra, ribs, all that. Then muscles also, muscles of the trunk and muscles of the limbs. So muscles also it will form. Okay, so it will give some bones and muscles, like that, something you can remember. Coming to the intermediate, what will happen to the intermediate? Something in the middle. So this will form genitourinary system, like the kidney, then the ovary, testes, all that they are talking about. Then you have that much you can remember. Then coming to the lateral plate mesoderm, this will form the diaphragm, that's a muscle, pericardial cavity, peritoneal cavity, body wall, right? Wall of everything it will form. Okay, this will become wall. This much you can remember. Bone, muscle, genitourinary, and outside it is wall. Okay, so this is the mesodermal derivatives. So this paraxial mesoderm, right, the one which is very near to the neural tube, that itself has that will form what is called as the somatomeres and somites, from which you are getting that axial skeleton, muscles, and all that. Okay, did that become too much? Shall we proceed with more embryology questions? So basically, and the next most important question of embryology is tongue. Okay, so what is the first most important question of embryology? NotoCord. Okay, NotoCord. And next important question is tongue. Development of tongue, they'll ask you 100 times. Now development of tongue, they have asked for 5 marks. They have asked development of tongue with nerve supply for 5 marks. And then they have asked the muscles of tongue develop from where? Is it from this? What do you think the answer is? It is from mesoderm. Don't worry, focus, we will explain everything. Okay, now some diagram they have shown here, we'll just look at it. So where is this? These are the somites they have shown. Very important word here for you currently, somites. So they have asked you the development of tongue, correct? So look at this tongue. We'll draw a nice pinky tongue. Okay, so this is the tip of the tongue. So we will say this is the oral part of the tongue. Okay, anterior 2/3. Oral part of the tongue. This is anterior 2/3, and this one is the posterior 1/3. This is the pharyngeal part of the tongue. Okay, so we will divide it into two parts. The oral part, that is in the front, this develops from the first pharyngeal arch. Okay, people, from the first pharyngeal arch you will get the anterior part of the tongue, and from the second, third, and fourth pharyngeal arches you will get the posterior part of the tongue. If you want more details, then we will explain now. So here, if you have the tongue again like this, the anterior part is developed from the first pharyngeal arch with swellings, the lingual swellings, and the tuberculum impar, some swellings. First pharyngeal arch, this will develop from the second, third, and fourth pharyngeal arches, that is from the hypobranchial eminence or copula of his, you will get this posterior 1/3 or the pharyngeal part of the tongue. Okay, from where does the muscles of the tongue develop? It develops from occipital myotomes. From where did myotomes come? We will come to that. So you remember around the around the notochord, you have the neural tube around, right? I mean, over the notochord, sorry, you have the neural tube, and either side of that neural tube, you have this intraembryonic mesoderm. So let me explain that. So here you have the neural tube, which is overlying this notochord, right? Guys, I'm just repeating what I have explained last time. So you have the paraxial mesoderm, then you have the intermediate mesoderm, and then you have the lateral mesoderm. From the paraxial, you had something called as the somatomere and somites. From the somites, you get the muscles of the tongue, right? In the somites only, there are again 3 parts actually. So you have the, too much it became, no? Anyways, you have something called as the myotome. From the myotome, muscles will develop. That is the tongue muscle will come from there. So remember, the paraxial was giving you the axial skeleton and muscles. So obviously the muscle has to come from this myotome, from the somite, okay, from the paraxial mesoderm, which is a part of the intraembryonic mesoderm. So paraxial mesoderm, somatomeres, somites. So somites have sclerotome, which is forming the bone, myotome in the middle, which is forming the muscles, and dermatome, which is forming the dermis of the skin. Here we are talking about the myotome, especially the occipital myotome, which will give you the muscle of the tongue. So muscles of tongue develop from occipital myotomes. Occipital myotomes are from somites. Somites are under what? Paraxial mesoderm, which is nothing but the intraembryonic mesoderm. So which is the nerve that supplies this muscle of tongue? That is from the hypoglossal nerve. Hypoglossal nerve is what? It's a cranial nerve, right? Which cranial nerve? 11? No, it is the 12th. 12th is the hypoglossal nerve. 11th is spinal accessory. What's wrong? Come on, say. 12th is the hypoglossal nerve. So musculature of tongue, hypoglossal nerve, 12th cranial nerve. Okay. Very good. Take a deep breath. Two more nerves to learn here. Why one for that tongue lingual nerve, glossopharyngeal nerve, superior laryngeal nerve. Wow, so much to learn. So let us just say this. The tongue is here. The front one is lingual nerve, which is a branch of the what? Mandibular nerve. Then you have chorda tympani, which is a branch of the facial nerve. Then you have the glossopharyngeal nerve, which is the nerve from the third arch. So actually, lingual nerve is from first arch, chorda tympani is from the second arch, glossopharyngeal is from third arch. From fourth arch also there is one nerve, superior laryngeal nerve it seems. Wow, so many nerves for the tongue. So somewhere here they are showing you the muscles of the tongue come from this occipital myotomes, the somites, okay, which are coming from the paraxial mesoderm, which is a part of the intraembryonic mesoderm, okay. So here you have the pharyngeal arches, where here pharyngeal arches are there here, nothing but the branchial arch also they call. From these arch you will get the tongue. From the anterior one, 2/3 you are getting from the first arch, and from the second, third and fourth you are getting the posterior 1/3 of the tongue, okay. So muscles of tongue, let's just zoom into this and see what they're trying to say. Tongue muscle served by the hypoglossal nerve. This is a cranial nerve. Which cranial nerve? Now you already learned 12th one. Very good. Just two more topics left, guys. Let me tell you. The questions that are left for now are pharyngeal arch and the derivatives and twins. Okay, these are the only two topics left. Don't worry, we'll cover it easily for you. Okay, people, so here is the question. First, branchial arch derivatives. That is nothing but pharyngeal arch derivatives that they want to know for 3 marks. So you have to just write very little. And then they have asked you the derivatives of second branchial arch also. Okay, so these two you have to answer now. Come on. So here they are showing you the arches. So the first pharyngeal arch, then the second, third, fourth, like that. So these are the arches. From this, what will you get? So you look at the frontal view of the embryo. 24-day embryo this is. So here you are able to see some arch, and this is the second arch, they're saying this one. So this is arch. From inside what you will have is pouch, right? Inside it is called a pouch. See, inside it will be called pouch. Wait, I'll zoom and show here. So this is the arch. From outside you will call it as arch. From inside this yellow thing, what are you calling? Inside this place is there, right? That is called as pouch. Okay. From outside that same place, which is pouch inside, from outside it's cleft. Okay, so the arch is there. Then you have the cleft. Inside the same thing is called as pouch. Right? So each of this has a nerve, cartilage and artery. So it has blood vessel, it has nerve and cartilage. Okay, each of these arch. They are asking you from the first pharyngeal arch, what will you get? From the second, what will you get? Third and fourth, they didn't ask, right? Only first and second they are asking. Above all this, they are showing some pharyngeal opening, okay? And down somewhere here, they are showing something called as foramen cecum. I'm seeing that bit. Where is that? Some foramen means opening, right? Cecum. So from the first arch, what will you get? Mandibular arch, okay? It is also called as mandibular arch. So you already saw it, right? In the front of that baby, frontal view. So you will get some skeletal derivatives, some muscular derivatives, some vascular element and nerve, okay? So let us look at everything that says mandible and we will be very happy with it, right? So you will get the mandibular nerve, mandibular process, sphenomandibular ligament, the mandible itself. So just touch your mandible, okay? And just see around it whatever is there that you say. So you will get mandible, some sphenomandibular ligament, maxilla also it is saying. You have some maxillary process and mandibular process. Muscles you will get what? Muscles of mastication, anterior belly of digastric, mylohyoid, tensor tympani, tensor palatii. What is the blood vessel? Maxillary artery, nerve is mandibular nerve. Okay, this is from the first arch. So basically they are saying the maxillary and the mandibular things you will get, correct? From the first arch. Meckel's cartilage, what is that? So I don't want to go into all this, you can read it if you want. Now let us see what we will get from second pharyngeal arch or second branchial arch. The second one they are calling it as the hyoid arch, okay? So it will give rise to, you touch hyoid, you touch your hyoid, okay? Not your thyroid. Above that you have the hyoid, correct? So that one. So stapes, styloid process of temporal bone. Stapes is inside your ear, right? Okay. Styloid process of temporal bone. Stylohyoid ligament. So some bones you should get, right? Then some muscles, the stapedius muscle, stylohyoid, posterior belly of digastric, auricular and muscles of facial expression. Then coming to nerve, the facial nerve. The facial nerve, the nerve of the second arch it is. So look at this blue one here, they're talking. So you have the stapes, styloid process, stylohyoid ligament, then the hyoid bone here, the upper part. So that is from the second arch, from this hyoid arch. So everything you understood and you have stored it in your brain now, correct? So just remember when you talk about the first arch, mandible, second arch, you can talk about the styloid process, stylohyoid ligament and the hyoid, upper part of hyoid, like that you can remember, okay? Everything around that only you will say, muscles and nerves, etc. See, did they ask you the derivatives of the pouch? No. Then don't write this. They have asked you the derivatives of the arch, okay? So not very difficult, isn't it? Super duper duper duper. Now we are going to the last question of embryology, and that is twins, 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 twins. How twins are formed? What are the types of twins? All that they're asking, okay? So mostly we will call it as multiple pregnancy, okay? Because twins means only two are there, correct? And twin, what are you saying multiple pregnancy? Either the conceptors are more than one, or there is more than one embryo, or there is more than one fetus itself, okay? So in one person. So that's what you can understand, multiple pregnancy. So if there are two, then only you will call it as twins. Two conceptus or two fetus or two embryos, you can remember based on which stage. And this is called as, at the same time, they will be there within the person. This is called as twinning, okay, twins. So you have monozygotic and dizygotic. Monozygotic means they have come from the same zygote. So there is one ova, one sperm, one ovum and one sperm. So these two fuse together to form zygote. This zygote will give rise to more than one person, right? Fetus. So this becomes monozygotic. So monozygotic means what? They have same genetic material. They are identical. So right. So they have the same father, same mother's. Same father, same mother. Only the same genetic material from the father and mother. Okay. That is monozygotic. Dizygotic means what? Dizygotic means the mother has two, she has given two right? And the father, there are two sperms which are independently fertilizing the two oocytes. See, the main thing you should understand is monozygotic and dizygotic. This much you understood, right? The main thing you should understand is something called as this chorionic and amniotic. Okay, that's what you have to understand. So let me explain here. So basically, if they are dizygotic twins, that means there are two ova, two sperms, etc. Right? So what happens? There is dichorionic, diamniotic. That means, look at this diagram here. So basically there are two placenta and there are two separate amniotic sacs, and these two twins will grow happily in their own world and they get their own nourishment. So this guy is not going to steal this guy's nourishment and this guy is not going to steal this guy's nourishment. So this is one of the best things, dizygotic twins. Okay, they won't be identical. Even the sex can be different. This can be a male, this can be a female, etc. So this is good. Okay, they don't share resources. Okay, but when it comes to monozygotic twins, they either can share resources or they can have separate. So that is what comes with monozygotic twins. Okay, so now look at this one here. Monozygotic, what I'm saying is they can have two placentas, two amniotic sacs, or they can have single placenta and single amniotic sac. That I will show you here. See this one. This is monochorionic and monoamniotic. In this case, what happens? They are in the same sac. These two can fuse and form what is like this, a conjoined twin, right? So this is really not that advised. Okay. Now look at this one here. They share a single placenta, but two different amniotic sacs. But here also the problem is each one fights for the resources. Okay, this guy can draw all the nutrition. In fact, he can draw the nutrition out of this guy also. Okay, so monozygotic is slightly more complicated. As long as they have everything separate, it is all good. Okay, that's why I put in green. And if they are sharing the same amniotic sac or the same placenta, it's not that nice. Okay. So they've asked you the embryological basis, so you can write the reasons that this happens. Mostly they say that the paternal age and the maternal age, right? And the history in the hereditary if they have twins, right? And then if it is they have more parity, that is they are delivered more than like 5 children, then the chances of twins coming in later is there. Okay, and it can be because of giving some IVF treatment, etc. by ovulation, by clomiphene citrate. If you're giving ovulation drug, right, it can release a lot more of oocytes, correct? Then maternal weight also they are saying, maternal hormones also, okay? So basically there is something called as TTTS and TRAP, etc. These are the syndromes where there is this twins, they kind of don't let the other twin get nutrition kind of things. Okay, also the delivery of these twins also is one concern for the doctors, isn't it? I think that much is enough, people. Okay, so this much if you know, I think this much is enough. Okay, for embryology to pass. Embryology part you will be able to clear. Okay, top priority questions embryology from the last 5 years previous year questions we have covered from anatomy final theory paper. Okay, bye bye.